Glycolysis is a series of reactions that break down sugar to provide ATP, which occurs in the cytosol of the cell. It converts sugar like glucose and breaks down into 3 carbon molecules like pyruvate. It needs investment of 2 ATP in order to work, and at the end, it generates 4 ATP for net profit. Glycolysis involves 4 main key events: phosphorylation, lysis, oxidation, and ATP formation. The first step of glycolysis is phosphorylation. The glucose will phosphorylate by 2 molecules of ATP, which make molecules less stable and more reactive and also prevent diffusion. In this step, ATP molecules is consumed and also produce 6 carbon sugar biphosphate. The next step is called lysis. The 6 carbon sugar biphosphate will split into 2 triose phosphate. After it splits into 2 triose phosphate, hydrogen atoms are removed from each of the 3 carbon sugar phosphate and transferred to the coenzyme NAD to form NADH. Some of the energy released from the sugar will immediately synthesize with ADP to ATP. At the end, the 6 carbon glucose has been broken down into 2 molecules of pyruvate. Glycolysis occurs in the cytosol, which means it is an anaerobic process and does not require oxygen. And because it involves oxidation, it will cause hydrogen reduce.
When pyruvate is produced from the glycolysis, which has been produced in the cytoplasm, the link reaction is the pyruvate is transferred from the cytosol into the mitochondrial matrix by carrier proteins on the mitochondrial membrane. And the pyruvate will lose a carbon atom. It is a decarboxylation, which forms a carbon dioxide molecule. Then it will have the 2 carbon compound, then forms an acetyl group. When it loses the hydrogen atom via oxidation, which is the NAD is reduced to NADH plus H. Finally, the acetyl compound then combines with the coenzyme A to form acetyl coenzyme A group. Actually, the link reaction is from a pyruvate convert to the acetyl group. And we have mentioned the mitochondrial matrix in the link reaction, so we should have some understanding with the mitochondrial matrix. In the mitochondria, the matrix is the space within the inner membrane. The mitochondrial matrix contains the mitochondria's DNA, ribosomes, soluble enzyme, and others molecules, and the enzymes is in the matrix promote the reaction responsible for the production of the ATP. ATP is the energy, so such as the citric acid cycle and the oxidation of the pyruvate and the beta oxidation of the fatty acid.
The second stage of aerobic respiration is the KREBS cycle, which occurs within the matrix of the mitochondria. A 2-compound fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the KREBS cycle, and the coenzyme A is released and can return to the link reaction to form another molecule of acetyl-CoA. A 4-carbon molecule is the starting material, and a 6-carbon molecule called citrate is made. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to the NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced as well. As a result of these reactions, a 4-carbon molecule is formed in the KREBS cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogen that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially react with acetyl-CoA. At the end, a 4-carbon molecule called oxalostate is formed, and it is the starting material of the Krebs cycle as well. As we know, each glucose molecule is broken down into two pyruvate molecules during the glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete two circuits to break down the two pyruvate molecules. Per glucose molecule, the Krebs cycle produces 4 carbon dioxide, 2 ATP, 6 NADH, and 2 FADH.
The electron transport chain is a process of transferring electrons between carriers in the memory of the crystal. The first thing I would like to introduce is the cytochromes. They are protein complexes playing the role of electron carriers in the process of ETC. Cytochrome 1 is responsible for depositing the electrons and H+ iron of NADH+. Cytochrome 2 is responsible for depositing the electrons and H+ irons of an FADH2, and the electron carriers will back to the Krebs cycle after depositing in the form of NADH+ and FAD. As receptor of electrons, the oxygen will grab the electrons from the last cytochrome in order to maintain the operation of the entire system. Here's what happens in the process. NADH+ deposits electron and H+ iron to the cytochrome 1. The H+ iron passes through the cytochrome to the intermemory space, while the electrons go to the next cytochrome. Since the electrons lose energy as they pass on ETC, some of this energy used up to make H+ iron from the matrix to the intermemory space of the mitochondria. Consequently, H+ gradient is developed. Oxygen is the terminal electron receptor. Water will be formed by oxygen combining the H+ iron in the matrix after electron exits cytochrome 3 in order to keep the H+ gradient. FADH2 also gives H+ iron and electrons to the ETC. The only difference between FADH2 and NADH is their electrons enter the ETC through different cytochromes. The animation shows what happens when FADH2 brings electron to the ETC. And this video could also be a summary of the process of ETC. The thing that we have to remember is that the whole point of ETC is preparing for chemiosmosis.
Chemiosmosis. In chemiosmosis, protons diffuse through ATP synthesis to generate ATP. The mechanism is to couple the release of energy by oxidation to ATP production remains a mystery for many years, but is now known to be chemiosmosis. This happens in the inner mitochondrial membrane is called chemiosmosis because a chemical substance moves across the membrane down the concentration gradient. This releases the energy needed for the enzyme ATP synthesis to make ATP. The main steps in the process are as follows. NADH plus H supplies pair of hydrogen atoms to the first carrier in the chain, with the NAD returning to the matrix. The hydrogen atoms are split to release two electrons, which pass from carrier to carrier in the chain. Energy is released as the electron pass from carrier to carrier, and 3 of these uses energy to transfer proton across the inner mitochondrial membrane from the matrix to the intermembrane space. As the electron continues to follow along the chain and more protons are pumped across the inner mitochondrial membrane, a concentration gradient of proton builds up. This proton gradient is a store of potential energy. To allow electrons to continue to follow, they must be transferred to a terminal electron acceptor at the end of the chain. In aerobic respiration, this is oxygen, which briefly becomes O2, but then combine with 2 H ions from the matrix to become water. Protons pass back from the intermembrane space to the matrix through ATP synthesis. As they are moving down the concentration gradient, energy is released, and this is used by ATP synthesis to phosphorylate ADP.